webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience for brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We are focused on understanding the molecular underpinnings of diseases such as PTSD and TBI to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk you through some logistics. This program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and website. The presentation will be approximately 40 minutes in length to allow for question and answer after the presentation. Please use the Q&A button in the Google Hangouts page to post a question. Today our speaker is Dr. Thomas Steckler. Dr. Steckler obtained his MD from the Free University of Berlin, then moved on to the MRC Neurochemical Pathology Unit in Newcastle, became group leader at the Max Planck Institute for Psychiatry in Munich, and eventually habilitated in biological psychology at the Heinrich-Heine University in Germany. In 2000, Dr. Steckler joined the Neuroscience Drug Discovery Group of Janssen R&D, where he has held various roles over the last 15 years. In 2015, Dr. Steckler moved to the Bioresearch Quality Organization of Janssen. Dr. Steckler is also principal editor of Psychopharmacology, is a member of numerous editorial boards, and is active in various international neuroscience societies. In this webinar, Dr. Steckler will discuss the factors underlying the lack of reproducibility in research, and will address ways to improve data reproducibility, robustness, and data quality using a forward-looking approach. It is my honor to introduce Dr. Steckler.
Yeah, hi everybody, and thanks for having me here today. It's a bit unusual for me because I've never given a webinar before, so bear with me. But I try my best to guide you through my slides and my talk, really. So let's start and assume that you are a scientist in a pharmaceutical company and you're told that you should develop a new anxiolytic compound, so a compound to treat anxiety disorders in patients. Ideally with a new mechanism of action because you want to have something different and something that works better than what is already on the market. What you possibly want to do is first you do a literature search. So you look at a number of keywords, and anxiety will be one of those. And you come across a number of publications. And take these three here that were published already some years ago in Nature Genetics, head-to-head. 3 papers, all 3 dealing with a certain receptor type, which is called corticotropin releasing hormone receptor 2, or CRH2 for short. And as you can see, at least 2 of those publications deal with anxiety. One talks about anxiety-like behavior, and one talks about anxiolytic role of that receptor. So it could be quite interesting receptor for your purpose to find the target for a new drug. So you look at the first paper. And what you see here is that they generated mice that lack a functional CRH2 receptor, a knockout animal. And they tested those animals on a certain behavioral test called the elevated plasmase. Elevated plasmase is nothing else than just a sort of a cross. So it's a maze with 4 arms that is elevated, hanging in the air, if you like. And 2 of those arms are open space, and 2 of them are covered with walls. And an animal will prefer these covered arms of the maze if it's supposed to be more anxious. So the more anxious the animal is, the more time it will spend in the enclosed arms, and the less entries it will make in the open arms. So that's the philosophy of the elevated plasmase. Now, as you can see here, there's the data. These animals were generated on a mixed black 629 SV background, the standard background for generating knockout animals, certainly at that time. And what you can see is that mutant animals that lack a functional CRH2 receptor spend less time on the open arms compared to wild-type animals, and they have less entries into the open arms, which would suggest that these animals indeed could be more anxious compared to wild-type normal control animals. If you take this in males and females, you see in both genders you have the same effect. So indeed, this might be an interesting target for you to develop a new compound, and what it might mean is that you develop a CRH2 agonist if you want to reduce anxiety in patients. So, well done, you're on your way. You go to the next publication. Also a CRH2 receptor knockout, same background strain, same test, the elevated plasmase. Again, males and females were tested. In that case, it was a bit more complex. They did not just test the knockout animals, but they also tested heterozygous mice, so indicated by plus and minus here, which would still carry one functional gene and should express a certain level of that receptor still. Now, what you can see is when you look at the males, again, you see that the knockout animals, they have less visits of the open arms compared to the wild-type animals, meaning, again, that possibly these animals could be more anxious. So, again, it backs up your hypothesis that you would want to have a CRH2 receptor to agonist to treat anxiety. However, when you look at the females, it's different already. What you see is there's no effect of your knockout whatsoever when it comes to number of open arm visits. So this might mean that there's a sex-specific effect. Now, given that in anxiety disorders you have primarily females being affected, this might then not be the optimal target. So you start to doubt a bit. And now you go to this third publication. In that case, they only tested males. But again, they used the elevated plasmase to test their knockout mice. Here you see the knockout being depicted with a white bar instead of the black bar as in the other two studies. But the key message here is that in this particular study, there was no difference whatsoever between knockout and wild-type animals in terms of open arm entries or the time spent on the open arms, which then would suggest, based on those data, these animals do not have any changes in anxiety-related behavior. Now, what are you doing then in that case? You have 3 different outcomes in 3 different studies published head-to-head in the same journal. You yourself start to fall in despair because you're at a loss whether or not to proceed with this target. And unfortunately, that is a situation we encounter again and again when we scan the literature, and not only when we scan the literature, but also when we try to reproduce data that have been published in the literature. There's a heterogeneity in results. And that problem is neatly illustrated by a publication that came from the group of Bayer a couple of years ago. They, in fact, tried to reproduce what's in the literature in a number of in-house studies. And as you can see here, they succeeded to reproduce those data in only a fraction of the in-house experiments. Really reproduced were about 20% of the data, and partially reproduced was possibly another 10%. But the majority of the studies were not reproducible. This is not in neuroscience, this is in the fields of oncology, women's health, and cardiovascular function, pointing to the fact it is not something specific to a certain area of research, but in fact is a general issue. Now, we, I think, already agree there is an issue here, and there could be different factors that come into play. A nice example for that is a study that came from Fanelli and was published in 2009. He did a survey among scientists about what could be wrong in terms of their own publications. And interestingly enough, about 2% of the respondents reported that they in fact had fabricated or falsified some data themselves in the past. Even more interesting was that more than 30% of the people said, well, we have done some other iffy things with our data sets in the past. And even more interesting was that when they were asked, what do you think about your neighbor next door in the other lab, more than 70% thought that there was some questionable research practices going on by their colleagues. So, so much about trust amongst peers. Now, the issues that you can face when you start to look at non-reproducible data, and in particular data that would be falsified or fraudulent could be pretty serious. Obviously, you can have retraction of papers, but of course there could be also huge costs involved. The costs of the investigation of research misconduct itself, there could be fines associated with it, but from a drug development perspective, of course, it is a disaster if there would be issues with your patents. There could be delays of compounds going to the market, or there could also be the need to withdraw compounds from the market. So a lot of issues at hand here. To avoid those issues, what you try to have is quality data, and strongly believe that to have quality data, you need to have quality tests and assays. And we, in the next couple of minutes, will deal with those in greater detail. I will use the area of neuroscience to exemplify what I'm talking about, and also specifically home in on in vivo studies. But of course, what I'm saying, I think is generally applicable to numerous areas of research. So what you want to have is validated methods and authenticated tools, so that you really use what you think you use. You want to have reliable methods and robust procedures. You want to have relevant results, standardized measures, and you want, last but not least, have objective studies that are free of bias. And we talk about all of that now. Now let's start about quality data and quality tools. In the neuroscience in vivo world, but in vivo in general, I think this all starts with having quality animals at hand. And we use the example of the CRH2 knockout to start with. So home in a bit on the situations with mouse mutants in general. This is an interesting set of data that was published last year in Nature, where they looked at close to 5,000 different mouse mutants that were published in the literature. And they looked at two things here. One was, where did these animals come from? And secondly, how often were these mutant mice generated? And the first important message here is that only a fraction of the animals really came from mouse repositories. Mouse repositories that are set up either at universities, or that could also be from other types of organizations like the Jackson Labs. The majority of animals were generated in small research groups and then bred and used by these teams. The other interesting outcome here was that only a relatively small percentage of these mice were unique. Many of these mutations were remade a couple of times, partly due to the fact that it is difficult to get access to animals that are generated by certain groups, and these groups might be hesitant to share these animals, at least initially, to also exploit these animals themselves. So you have a number of animals made more than once, 3 times, or even more frequent. And of course, there could be differences in the genetic background of these animals. There could be also differences in the exact ways these mutations were made, all of which will contribute to potential differences in the phenotypes of these mice. Now, to avoid this, as I said before, you could go and adhere to some infrastructures, standardized infrastructures when it comes to mouse mutant generation. To give you an example of how we achieved this in a consortium where we participate, which is called EU AIMS. It's an IMI consortium that focuses on autism spectrum disorders, and where we also test animal models of autism. So what we did in that case here was that we selected a number of mouse models that came from the academic partners from that consortium based on a number of predefined criteria. These animals were then brought to a mouse resource facility, which was a commercial breeder. All these animals were re-derived, and then that breeder would distribute either breeding pairs or experimental animals to the different partners in the consortium. And those partners would then share a mouse phenotyping effort, looking at types of behavior or other measures that would be of relevance for animal models of autism. And I think that's possibly one way forward to reduce heterogeneity in findings between labs in mouse mutants. Because what happens, or what is a risk you face if you do not adhere to these principles? One risk you have here is that you might misidentify your animals. Now you could argue this is not happening to me because I know quite well what my mice are about. However, it has been shown that of about 200 mouse lines that were deposited in these mouse mutants research centers, about 16% did not really match the phenotypic data that were initially described by the researchers. Also, there could be non-specificity of your mutation. Again, it has been shown that of more than 400 mice that were submitted to Janssen labs or the mouse research and research centers, about 15% did carry mutations other than what had been specified, or still carry genetic markers that were not intended to be part of that line anymore. There could also be decoupling of genetic markers, another risk you're running in your mouse lines. If you use a genetic marker to, for instance, mark your mutation and you breed for that marker, you have to make sure that it is still linked to the mutated gene. But what can happen is that there is a decoupling between marker and the genetic mutation. So you might still breed for your marker, but your mutation is gone. Also, you might have difficulties to control for genetic drift if you just generate these mice over time. And then there could, of course, be secondary non-specific effects coming into play, like pathogens that might be present in your facilities. All of these factors should be well controlled in these mouse research centers. Last but not least, you have to breed your animals in sufficient numbers to come to meaningful results. And again, that might be tricky if you have limited capacity to do so. And if you have animals that you can order from Janssen labs or other facilities, at least you should end up with numbers that are meaningful to run your experiments. But what can happen if you don't do that? So take the example of misidentification. Misidentification can, for instance, happen because you do not have all the information that you need to identify your animal. And this is exemplified here. Take the example of the C57 Black 6 mouse. It's a strain that is frequently used to generate mutant mice. So it serves as a background strain for these animals. Now, as you see, there is one Black 6 mouse that has an N and one that has a J at the end. N is a Charles River mouse, J comes from the Janssen lab. These animals differ, even though they are pretty identically looking. The N strain line carries a gene which is called RD8. RD stands for retinal degeneration. And as a consequence of that, these animals show retinal pathology developing as the animal matures. This is not the case with the J line that is free of that retinal degeneration gene. On the other hand, this animal seems to be protected against diet-induced obesity. So if you would feed these animals with a high-fat diet, you might find that these animals still stay pretty lean over time, whereas the N line would gain weight substantially. This can have consequences depending on the exact question you want to ask. If you have, for instance, a behavioral paradigm where you look at a task that requires a good vision, that could be, for instance, a maze procedure, as I've mentioned at the beginning of my presentation, you might see that there could be issues with an animal that is genetically compromised in visual acuity. Of course, these animals also differ in a number of additional genetic loci. I think 11 have been identified to differ between these lines. So there could be subtle differences which have consequences on your behavior. And depending on how you breed your mouse mutant line, you might introduce different phenotypes, depending on whether you breed or cross these animals with a black 6N or with a black 6J line. But this lack of information is not, again, limited to mouse mutants. It's not limited to behavioral studies and to neuroscience. In fact, it's an issue that we are facing in a lot of areas in research. And this is exemplified here with a study that was published in 2014, where they looked at the information that's presented in the literature. And what they've done here is they selected a number of articles published in PubMed in 2013, and they then scanned the method section for the information available. And what you can see here, lo and behold, is that more than 50% of the information that would be really required to reproduce a study, it wasn't present. That could go for antibodies, that could go for cell lines, that could go, of course, also go for mouse mutants in that respect. Yeah? And having realized that, there have been some initiatives trying to rectify that result, and one of those is the Resource Identification Initiative. I think it's a very interesting initiative that tries to apply unique identifiers to the different materials and animals used in the research. I've exemplified that here for rats. For the Vista rats, it's an outbred strain for rats used frequently in animal research. And as you can see here, there are some sublines available. The Vista rat, Vista Ulster, for instance, WU, or the Vista rat Hanover, abbreviated with HAN. And as you can also see, these animals can be obtained from various sources, commercial suppliers and breeders, like Charles River, for instance, like Taconic. There might be subtle differences between the different sublines. There could be subtle differences between animals coming from different breeders, and pretty much along the same lines as I explained it for the Black 6 mouse. You would really like to know what exactly you're testing when you're testing these type of rats. If you just have a Vista rat, this information might not be sufficient. But if you would go for these research resource identifiers, the RRIDs, you can see there are unique IDs that are available depending on what subline you're talking about, and also exactly what supplier these animals come from. Now, this system is still in development, but it's already used in a number of journals, and I think it's an excellent approach to really get a better handle on what exactly we are using in our research. It's also available for antibodies and cell lines and you name it. The second point, of course, was the insufficient number of animals I've adhered to when I talked about the breeding of mutant mice. And that also is an important issue we need to adhere to it, because the power of a study is related to the number of animals you're using. That's exemplified here with this hypothetical graph. You see that as the number of animals increase, there's an increased power in your study. The power is nothing else than the chance to correctly detect a difference between groups. So you could argue the more animals you use, the higher is the power of your study, and the better is the probability that you detect what is in fact there. Conversely, you could turn the curve around and you could argue that if you do not use enough animals, the chances of wasting animals increase, or with more animals you have a decreasing chance of wasting animals. Yeah, so what you want to have is a sufficient number of animals. That has been realized, in fact, over the last years, and the funders now start to demand that there are sufficient number of animals. I've just highlighted this here from some recent discussions that took place in the UK, but also, of course, NIH started now to request that there are statistical analysis of the power of your studies and the animals needed for your studies before you start applying for grants. Now, of course, you could argue why then we could use as many animals as we would like. That, of course, goes against the principles of the 3 R's, which means reduction, refinement, and replacement. So people think rather the other way, they should use as low number of animals as possible. I would argue we should go for the 4 R's, really. That means you should also consider the right N. You should not use more animals than necessary in your studies, but you should also use sufficient number of animals to have reliable data. That this is not the case in many studies is exemplified here in a review from Button and colleagues that looked at the power of again studies published in the area of neuroscience. And as you can see here, the vast majority of studies that were published and looked at are actually underpowered. So close to 60% have a low power of below 0.3, really. Yeah. Conversely, there's only a fraction of studies, about 15% here, that have a very high power. And again, you possibly do not need to go that extreme. But if you would have an intermediate power in your study, I think it would help you enormously to get more reliable data sets. You also want to have relevant data. That's another aspect you need to consider when you start your experiments. So you have to ask yourself the question whether the tests you're using really measures what you intend to do. And exemplify that here, again, with an animal behavioral study, in that case, passive avoidance, and exemplifies this here with this little cartoon. You see there's a 2-compartment box with a mouse in. One is brightly illuminated, one is dark. And the idea is that an animal that is anxious would try to escape to the dark compartment. And rodents really prefer the dark compartment. So it's a bit the same philosophy, if you like, as for the elevator plus maze before. The memory component comes in because as soon as the animal enters the dark compartment, it gets an electric foot shock. And it learns to avoid the dark compartment. So just the time the animal stays in the light compartment before it transits to the dark compartments and is taken as a measure of how well the animal remembers over time. It's a test that has been extremely popular and partly still is in the attempt to find cognition-enhancing drugs, and particular to look for compounds that could be of utility for instance, Alzheimer's disease. And a model that has been used frequently in the past was a so-called scopolamine-induced passive avoidance deficit. So what you do here is you treat these animals with scopolamine, which is an antimuscarinic drug that is known to impair cognitive function. And if you do that, you can see that animals are tested in the passive avoidance paradigm will show so-called memory deficits. So they would just transit to the dark side of the compartment, even though they had experienced a foot shock before. Now, this paradigm has been used quite frequently for testing of cognition-enhancing drugs because it's fast, it's easily applicable, and it seems to produce reasonably reliable and reproducible results. However, when you look at the translation of these results to the clinic, it fares rather poor. And this is exemplified here in this little graph. I've just taken these numbers from a review that already has been published quite some years ago by Martin Sauter and colleagues. And he looked at avoidance tests and how well they fare in predicting clinical efficacy. And at that time, he included in totally 29 studies using the scopolamine model in passive avoidance, and which also were used to test compounds. And 24 different so-called cognition-enhancing compounds showed efficacy in the model at that time. However, when you look at how many compounds transitioned and made it into the clinic, it was 0. That means that this paradigm possibly is not the best to predict cognition-enhancing effects in Alzheimer's patients from these animals. It just illustrates that you have to have the right model. Still, there are publications using the passive avoidance paradigm to make these type of predictions today. Because what you want to have to make predictions is that you have a relevant model, and relevance, I believe, is driven by the generalizability of your tests. So meaning you have to have a model that using a sample group would then be predictive for the population at whole. That could be within the species itself, but could also be across species in a type of translational approach. Now, having realized this, NIH already questioned the predictability of a number of paradigms, because a lot of behavioral studies are limited in predictability simply because they neglect a couple of factors. One very important factor here is sex. I've shown you these studies initially with the CH2 knockout mice tested in the elevated plasmids, and you noted there were 2 of those studies that tested the knockout in both males and females. And this I would call already a very advanced approach. There was one study that only used males, and I think this has been, and to a large extent still is, the standard, unfortunately, in current research practice, because there could be quite dramatic differences in gender effects. There was this one CRH paper I've shown you where you did see effects in males but not in females, already pinpointing to that effect. But of course it could be even more pronounced, and I've shown you this here is an example of an imaginative experiment. I've actually stolen that slide here from Larry Tabak from NIH. So supposedly you test a new treatment and you want to see whether it has an impact on disease. When you look at the effects of the treatment across the population, you do not see any effects at all. This is the aggregated groups here. But if you would only select males, what you see here is that there is quite a difference between your treatment and your control in male animals. And conversely, if you test this treatment in your females, you see the opposite effects. So that illustrates that gender could in fact be of major influence. You could argue, no, well, that would not happen. This is something that is really imaginative. But I'll show you some real life example here. This is a study where they looked at a compound, a PARP1 inhibitor, to see whether that would have an effect on ischemic brain damage. And so what they looked at here was at infarct size. And what you can see here is that if that compound was used in males, it clearly reduced the relative infarct size, so meaning it had some beneficial effects. However, when they did the same in female animals, it had the opposite effect. This just highlights again the importance to test in both genders to be representative for your population and to also be more predictive than for the patient population. But there are other factors coming into play as well. We've already alluded to the strain, the background, for instance, that plays a role. We alluded to species that may play a role here as well. But of course, age could be another factor. The history of the animals, whether they had stress exposure somewhere or not, the way they were housed in an animal facility, all of that could play important roles in the outcome of your research and could contribute to irreproducibility of your data. So what can we learn then? Might be that we should take a slightly different approach, and I believe we can possibly learn from the area of toxicology, because in toxicology, rather standard, people adhere to some rules which I think make a lot of sense. One of those rules is that they test in both sexes, so they use males and they use females by default. But they also, as a rule, use different species. So they would not just test in a rodent, but they would also test in a non-rodent species. And the impact here is illustrated from a paper from Dixit and Bolsterly in 2007. So they looked at the prediction of toxicity for the human situation based on preclinical data. And you can see that in about 40% of cases it was correctly predicted when only rodents were tested. In about 60% of cases it was predicted when non-rodents were tested, but in more than 70% it was correctly predicted when 2 species were tested. So this clearly indicates that you can improve your predictability, your reliability of the data if you take the extra effort, and I would advocate strongly to do this, to test in different sexes and to ideally also test in different species to have more generalizable data. Now coming to the objectivity point. And I'd like to illustrate this with 2 little experiments here. Again, this is a behavioral study, and again it has to do with genetics. There have been in the past a lot of studies looking at inbreeding traits. So what you do here is you take a number of animals on a certain strain, for instance a mouse strain or a rat strain, and then you test these animals for a certain type of behavior, for instance, or any other phenotype. And then you segregate your animals, you breed them into, for instance, high performers, low performers. You cross those high or low performers, and you sort of segregate those animals for these traits. In this particular study here, they did this apparently for rats, and so they had some students, and these students were told, look, you have these 2 groups of animals. They have been bred for high cognitive function and low cognitive function. And these students were asked to test these animals in 2 paradigms. One was a maze paradigm again, a T maze, where the animals had to learn to either turn left or always turn right to get a food pellet. And the other was also a discrimination task, where the animals had to learn to either press a left or a right lever in an operant chamber to get a food pellet. And of course, these animals like doing this because they would like to get the pellet. So tests that animals learn reasonably well. And as you can see here on the data for the T maze on the left-hand side, you see that the animals that were bred for high performance outperformed indeed the dull animals. And the same was actually found in the operant paradigm. Animals that were bred as high performers outperformed these animals that needed less trials to learn the task. So in both studies, it was seen what was expected, namely that the animals bred for high cognitive function indeed performed better than the other animals. Now this is what really happened. In both cases, the experiment group was not the animal group, but was a student group. And so what they did here was that the experimenters just took some animals at random, standard laboratory rats, at random assigned them to 2 groups, a group of bright rats, a group of dull rats, and gave those animals to the students. And told these animals, look, you have these 2 groups of animals, and lo and behold, the students found what they expected, namely that the bright rats outperformed the dull rats. And this is what is called the investigator bias, and a very important aspect you need again to take into consideration when you run your experiments. This is not again limited to animal behavior, but something that is inherent as a factor that could confound your data in any type of experiment. And below I show you the 2 publications that reported on these data really. How can you avoid those investigator bias? You have to make sure that the experiment itself is blind to what is testing. And of course, you also need to ensure that the experimenter doesn't know the order in which these, in that case, animals were tested. So you would also like to randomize your study, because otherwise you will always run the risk unconsciously to see what you want to see. However, when you look at again what's in the literature, you see that only a fraction of studies report blinding of experiments, and only a fraction of studies report randomization, despite the importance of these factors. In fact, there has been a study assessing the effects of blinding and randomization and showed that the odds ratio was pretty high. So the likelihood that a study reporting positive results, if it has not been blinded and not been randomized compared to a study that had been blinded and randomized, was very high. Despite that, we still face that issue today. This is from a very recent publication from Marco Magliot's group from Edinburgh. He looked at the UK scene, and in that case he presented data from the 5 top-rated UK universities, and he looked at the publications from those universities and whether they reported randomization, blinding, whether they reported pre-specified in- and exclusion criteria, or sample size calculation to estimate the power of your studies. And you can see again the number of publications reporting this is pretty low, yeah, approaching really zero, in fact, for sample size calculation even. So this is not a really a good example on how to do it. You also would like to, of course, have a full picture on all the data that have been generated in the field. Supposedly, you would have had only the first publication on the CRH2 receptor, showing that you would have beneficial effects of, or you would have, or suggesting you have beneficial effects of a CRH2 agonism for anxiety treatment. You would have missed the other 2 publications, the one which was a bit more questionable and the other one which showed in essence negative results. However, when you look at the chance that publications get into journals, you see that primarily those studies are published that really show positive results and strong positive results. When it comes to no results, the majority of studies do not see the light of the day and end up in the drawer. In fact, that's why it's called the file drawer phenomenon. So another source of potential bias when you want to make your mind about a certain factor. Last but not least, you would like to know the truth. And this brings us to the last point of issues at hand and gets us back to data fabrication and falsifications. And a lot of you might be aware of the Recon Institute issue where data related to the stem cell field were falsified. And that had serious consequences for the whole institute and for the scientists involved. But there are other related issues that came up more recently, for instance, looking at peer review processes. And again, there could be fraud in that aspect as well. Springer has just retracted 64 articles from 10 different journals because there were issues with the peer review process and fraudulent behavior, people starting to review their own manuscripts in essence by setting up wrong email addresses and fake reviews. It's not an issue that is limited to the academic world. It can happen in industry as well. And the consequences can be pretty serious, including jail, as has recently been seen for researchers that fake data on vaccine development for HIV. Now you could argue, well, that cannot be really so much of an issue because we all are honest scientists. But however, if you look at studies that really looked into that issue, they would argue that misconduct could potentially be accountable for the majority of retractions in the literature. And in that particular study here that was published in PNAS in 2012, close to 70% were suggested to be retractions due to misconduct. And of course, if you then see that the number of retractions is also an increase, you could argue that there is a risk that misconduct is also on the rise. The consequences of misconduct can be substantial for individuals. That could mean that you lose your job, that awards are revoked, there's a lawsuit pending. Of course, the integrity of the person is potentially destroyed. There could be capital issues as a consequence, a lawsuit again, legal costs involved, investigation costs, I've mentioned those before, loss of grant money. These are huge numbers. I've mentioned the $500,000 US for an investigation already, but even it has been estimated just the annual costs for these type of investigations amount to about $100 million per year in the US, and these were 2010 figures. Irreproducible research in the US alone has been estimated to amount to close to 28 billion per year. If you are, again, in pharmaceutical industry, you risk that your brand is damaged, your reputation is damaged, you might again have to retract compounds, but also you lose talents because people might not want to work with you anymore, leave your company, or you might have difficulties attract new talent, and of course you lose sales. And then, of course, there's a human factor as well. If you want to develop, for instance, a compound, the patient suffers. You risk misdiagnosis, mistreatment, you misguide research of your peers, and there's loss of time. And just to illustrate this point, as an example, there has been a case where an anesthesiologist fabricated data in at least 21 papers, many of which were published in a journal called Anesthesia and Analgesia, and it was the editor-in-chief of that journal that was quoted. It's not evidence-based data here, but I think there's an important message here, thinking that about millions of patients worldwide could have suffered because of the misleading data from that one scientist, potentially leading to mistreatment and post-operative pain management. So you can see how serious these type of fake papers could impact on patients as well. But of course, there's also the error factor that comes into play when it comes to retraction, even though, at least in that particular study, it's estimated to play a relatively small role only. Nevertheless, errors can happen, mostly leading to corrections rather than full paper retractions. But if you look at the corrections again, you can see that often there is quite an extensive time lapse between the initial publication and the correction. In that case, which I'm highlighting here, it's 2 years. So this was a really frank error, and the authors corrected that, and everything was fine. But of course, to be able to correct, you need to also know exactly what you have done and what your data looked like when you published. And this brings me really to the last point of my presentation, because it's all about your data itself. Yeah, you could argue, well, I've done my study, and why should I bother about my data? It's published now, so I can just get rid of it. However, in case you might spot an error in your publication or in your dataset, or someone else does, which is possibly even worse, you want to be able to get back to your data. You want to validate what you have done. You want to look at your data again. You possibly want to also use your data again. You want to reanalyze your data to test a new hypothesis. You also might be asked to prove your data. This comes into play, for instance, when you get an audit in your lab. And there are some founders that think along those lines now, auditing labs which are funded by them. Could be also, certainly when it comes to industry, that there are patent disputes, and you want to go back to your data to prove what you have done and what you have claimed in your patents. You might also be asked to provide your data simply for contractual obligations, because, for instance, you have a collaboration with industry, and they ask you for the raw data. Or more and more, what you see is that publishers, journals would like to also get access to your raw data as part of the normal publication process. So these raw data are important. You need to store them securely, and you need to be able to retrieve them. Of course, there's also the fact that for some funders, for instance, they ask you to retain your data as part of the contract you get with them. At least 3 years, for instance, specified by the NIH grants policy statement. So you have to make sure all of your data are available. Now, as I said before, these things have to be adhered to everywhere, whether it's in academia, whether it is in industry. I just want to finish up by telling you how we at Janssen deal with that. To give you an example of our own efforts, because we wouldn't want to finger point, we want to also keep our own shop proper. And we have what we are calling the Drug Discovery Integrity Initiative. It's a sort of quality maintenance system where we have a quality program. So we have a quality assurance program and metrics. Here we do spot checks of the data that are generated in-house. Key data, so obviously we don't have the capacity to look at all the data that are generated. And particularly we're looking for the traceability of this data. So can we find the raw data back? Are those data securely stored? Can we reconstruct from these raw data the data that are presented by our scientists? And we will look at the integrity of the data. Are these data free of bias? Are there not any issues with the data in terms of leaving out certain data, having predefined outliers? Are these studies powered sufficiently? Are they blinded? And all these points I've made before. We also try to change the culture and to have a positive effect on the way our scientists behave. So we have information campaigns, we have poster campaigns, for instance, where we remind people on the importance of proper quality in a preclinical area and non-regulated area. And we also have communications by our senior management to the organization. Moreover, we have training sessions in the good research practice as part of our routine training in-house. We also did a gap analysis. This is a joint effort. This cannot be done just by a quality organization on its own or by other professionals. It's a joint effort between quality and scientists to really identify any issues at hand. We have formed multidisciplinary teams that really try to come up with best practice and solutions to tackle any gaps we see. Those could be different people involved, could be IT, legal, communication, statisticians, quality people, the researchers again, and to make proposals for solutions to improve. So it's a continuous improvement process, really. We have implemented what we thought are best practices. And of course, we want to also prove that these are efficacious steps. So we feed those data generated with these practices again into our QA program and see whether we in fact are efficacious. So it's a continuous process, really. That's our own internal efforts. So to close then, what is quality research? I think, or I hope I did show you that what you want to have is solid processes to prevent bias. You want to have validated materials, authenticated materials. You want to know exactly what you're using. You want to have standardized processes, appropriate controls and baselines. You want to have pre-specified in- and exclusion criteria, start and end points. You want to use randomization and blinding as much as possible. You want to have data integrity, meaning that you need, again, to pre-specify your outliers up front, not post hoc. You want to report your results accurately to really reflect the data you have generated. You need to have data that are securely stored. You want to have consistency in your databases from the raw data to the processed data to the analyzed data and what you eventually report. And you want to have consistency also between your experiments. You want to have replicatable data across your studies. You want to have solid statistics at hand, so you need to have appropriately powered studies, and you want to have ideally upfront statistical advice. Data have to be relevant, so they should be generalizable. You want to have data in both males and females. You want ideally have data that are generated in different genetic backgrounds when it comes to, for instance, tests of mouse mutants. You want to look at age effects depending on exactly what questions you want to address, of course. And you want to have data that are fit for the intended use. So for instance, if you want to run a study for cognition-enhancing drugs, it might not be the best idea to use a passive avoidance paradigm. Last but not least, you want to have transparency. So your data need to be traceable when it comes to storage, retrieval, and reconstructability. And you would want to do full disclosure. So you do not just show the positive data, but you want to include all your data, which also means the negative data, not just the 2 experiments that worked out of the 20 you did. And that concludes my presentation. Thank you very much.
Well, Dr. Steckler, thank you very much for a wonderful presentation. I'd like to open up the community to submit any questions at this time. We have a couple of minutes left. And just remind everyone that you can watch this webinar on the Cohen Veterans Bioscience YouTube channel. And again, you can contact us at any time with questions about this webinar, others. So it doesn't appear to be any questions at this time. So Dr. Steckler, thank you so much on behalf of Dr. Magali Haas and Cohen Veterans Bioscience for just a wonderful presentation, very highly informative. It's tackling a huge challenge in the field at this time. So thank you so much for your time.
You're welcome. Bye.
Goodbye.